We are now going to begin the second half of Unit 2. So this is Part A for Unit 2, Part 2 PowerPoint. So we've already discussed within a cell, and we've discussed between cell generations. So this last half of Unit 2 is going to talk about between cells of the same generation. This is essentially horizontal gene transfer, where you have a donor cell, that means it's giving something up or some genetic material, and then contributing part of its genome to a recipient cell. There are three types of horizontal gene transfer that we're going to cover. The first one is known as transformation. The second one is known as conjugation, which I have mentioned before when we've talked about the pili. And the final one that we'll talk about is transduction. So let's begin. Let's begin by taking a look at transformation. So transformation, Griffith had an experiment that pretty much defined and explained transformation type of horizontal gene transfer. For this particular type of event, I would like you to think of the word dead or dead DNA from the environment. So in all three types of horizontal gene transfer, it's again, there's a donor cell giving up some of its genome, so some genetic material, and giving it to a recipient. So let's take a look at this experiment. You have pathogenic bacteria that's been injected into a rat. The rat has died as a result. This is color-coded as red. Then you have harmless bacteria. When something is harmless, that means it does not cause harm, and therefore this rat did not die. Then we had heat-killed pathogenic bacteria. So I want you to think of a time when there is a flood or some natural disaster. People are told to essentially boil the water to make it clean and get all of the pathogens and bacteria out of it. So here they took something that was pathogenic and essentially heat-killed it or sterilized it. Because it was heat-killed, when it was injected into the rat did not die. So in the last case, we have a mix. We have the harmless bacteria and the heat-killed one. So let's go back to the beginning of transformation and look at what grew on the actual Petri plates. For the pathogenic, it's color-coded red. It killed the mouse, and you see colonies of that particular pathogenic bacteria that's been grown by isolating from the dead mouse. Then you have the harmless one. The harmless strain is denoted by a purple color, and again, you have colonies of that purple harmless bacteria growing. When you have heat-killed bacteria, because you have killed it off, if you just compare the shapes of the regular pathogenic and the heat-killed, you could see that the shape in itself has changed. And then when you look at the Petri plate, nothing was actually isolated. There were no colonies because the bacteria was killed off with heat. Now when you take a look at the last one, where again you mix the harmless bacteria with the heat-killed one, so look at the pictures from before. Here's the red heat-killed pathogenic, and then the purple one, which is the harmless. Notice now on the plate you have both of them actually growing, except take a look at what happened to the heat-killed. It essentially now looks like the pathogenic bacteria from the original plate. So when transformation occurs, what's happening is it is picking up dead DNA from the environment and incorporating it into healthy DNA within that particular organism and essentially bringing it back to life. So scientifically what's happening is genetic alteration is occurring through this transformation when a cell is picking up DNA directly from its environment and then expressing it. The dead part is the fact that it is picking it up from the environment. And remember, of the original injection, that also included the heat-killed pathogenic bacteria. So here's another picture of transformation to make it a little bit more visual. You have dead donor cell DNA. So again, this is the donor. Notice that it is broken apart. So now the DNA is in the surrounding environment. There is a live recipient that actually takes in. So if you look at the color here, that DNA from the dead one is red. So that DNA is now entering a live recipient cell. So here's the terms of donor and recipient. Once it incorporates it into its genome, from the environment, what happens is it starts disintegrating the donor strand and then incorporates it into the genome of the original strand. This is known as a transformed recombinant cell. So when you have transformation, you have a recombinant cell or a transformed cell. And again, this is picking up dead DNA from the environment and then incorporating it into a live recipient. The second type of horizontal gene transfer we're going to talk about is conjugation. In conjugation, again, you have this pili. Here it is right here. And this pili is almost like a tube. In lecture, I tend to talk about when you have an airplane in the military and a second one shows up that's a fueler jet. They pull that hose to mid-air fuel the other plane. So when this pili attaches the bacteria, what ends up happening is genetic material can pass between those two bacteria. One is the donor, which is giving up the DNA. The other one is the recipient. There are only two types of material that we're going to talk about, or genetic material that can pass. One is F plasmid, because it is the plasmid that goes through. If you think back to Unit 1, we discussed this. So the F plasmid is for fertility. And the second type we're going to talk about is the R plasmid. And we have almost an entire unit that is dealing with the R plasmid because R stands for resistance. So the first thing you should think of is antibiotic resistance. This is conjugation contributes highly to antibiotic resistance in bacteria. And I do want to point out, since you need to know this, of the three types of horizontal gene transfer that we talk about, conjugation by far is the least effective. And the reason it's the least effective is because this pili can become detached in the middle of the process of genetic material transferring. So it's hard to know what does or does not make it through, which is also why it causes resistance. So here's a visual representation. There's two types of conjugation, but essentially one is faster, one is slower, and they are a little bit similar. So here you have the bacterial chromosome, and here you have the little plasmid. This right here is your donor, and this right here is your recipient on the other side. What you should notice is that the two are attached, and they are attached by a pili or pilus. So what happens, if you take a look, this one is known as F positive. So if I asked you what type of plasmid this was, even if this wasn't written, you would know that it's an F plasmid, because remember, we are only going to deal with F and R. F is for fertility, R is for resistance. So this is F positive. That means it contains that F plasmid. This recipient is F minus, which means it doesn't contain that particular F plasmid. Through conjugation, this rolling circle mechanism, notice that the genetic material is passing from the donor to the recipient. And then if you look at the final picture, the donor remained an F positive, which means it did not give up its entire plasmid, just a portion of it, which then made the recipient also become F positive. So now both the donor as well as the recipient contain the F plasmid. The second concept of conjugation is known as HFR conjugation. And HFR is for high frequency. In this particular case, conjugation is occurring at 1,000 times the rate of regular conjugation. So, excuse me, this is the original picture from the previous page. Notice that plasmid becomes incorporated into this bacterial chromosome. So now this is essentially the genome. Then there's a pili. And again, through the pili, you have that rolling circle mechanism where some of this genome is transferring over to the recipient is sending it over, or the donor is sending it over to the recipient. And now both the donor as well as the recipient are going to have a portion of that genome passing over. And now you have this full recombination. And this again is HFR, high frequency recombination, making it at the rate of 1,000 times faster. In general, just a little piece of interesting information is it takes 100 minutes to transfer, so about an hour and a half, to transfer all of E. coli chromosomal DNA with the plasmid genes from the donor to the recipient. Finally, the last one that we're going to talk about is known as transduction. So this is the third type of horizontal gene transfer. And this one's a little bit unique and different as opposed to the other two because it also includes viruses. So there's a virus that infects and usually that it can infect and then can infect a bacterial cell. So this picture on the bottom here is one E. coli, so one rod of E. coli, and all of these little greenish blue things are bacteriophages. So the definition of a bacteriophage is a virus that can infect bacterial cells. So what happens is it's almost like a lunar landing, as I call it. So if you take a look at the first picture on the top left, what you see is the bacteriophage landing on the surface of the bacterial cell, and then it scooches down within the head or capsid of this virus, and our last unit is viruses, so this will be covered. Notice it's injecting its genetic material that's in the head of the virus into the bacteria. By definition, transduction is a type of horizontal gene transfer where you are transferring genetic material from one cell to another, but the means is a bacteriophage, which is in fact a virus. So there are two different phases that can occur. The first is lytic, and on the next page you'll see lysogenic. So let's take a look at the pictures here. Looking at number 1, attachment is the first step where the bacteriophage attaches itself to a bacteria. And again, remember, this bacteriophage is in fact a virus. This is the bacterial chromosome. We're not calling it a genome because nowhere in this picture do you see a plasmid. The second step is entry. Again, from the head of the bacteriophage, the genetic material of the virus is now injected into the bacteria. Next, the bacterial chromosome, which is depicted in purple, begins to degrade. And what is this green thing? This is the genetic material from the bacteriophage, which is again a virus. So now you have synthesis. Viruses are intracellular, non-essentially alive parasites. So therefore, this genetic material, or phage DNA, is inside of the bacteria. It begins to synthesize or propagate itself through the tools and the machinery of the bacteria. So once synthesis occurs, you have assembly. So inside of the bacteria, you should see that several new bacteriophages are now assembling. It's making a little army of itself. And the last step is release. So now all of this viral genetic material that is meshed together with the bacterial chromosome that was originally degraded is released into the environment so these phages can attach themselves to other bacteria in the surrounding area. This is the lytic phase. By lytic, it implies it occurs pretty quickly. So again, the bacteriophage attaches, then it enters the bacteria, then it degrades the bacterial chromosome, it starts synthesizing using the machinery of the bacteria, assembles its little army, and the last step is it gets released. The second stage is known as lysogenic. This one is a lot slower, or you can also say that it takes longer, and you'll see in a minute why that's the case. So if you take a look right here, you're 1, 2, if this were 3, 4, and 5, this picture right here is your lytic phase. So you have attachment, entry, it's synthesizing, assembling, and releases. But that isn't what occurs for lysogenic. There are a couple of steps in between. So let's add those steps so that everybody can see them. So what's happening in lysogenic is that the phage, when it enters and starts doing its synthesis and assembly, it doesn't immediately kill its host the way it did in the lytic phase. What happens is you have the bacterial chromosome. Here is the bacteriophage attaching. And again, you have entry of the genetic material. And instead of the bacterial chromosome breaking down, that doesn't occur. The phage DNA incorporates itself into the bacterial chromosome, which then starts doing this odd version of binary fission, so it's replicating and replicating and replicating, and eventually an environmental stressor comes in, which then causes it to start to degrade the genetic material, as you can see right here, and then we have our entire assembly and synthesis occurring, and at that point you have a release as a final step. So the difference between lytic and lysogenic, lytic immediately kills the host by breaking down the bacterial chromosome and then assembling an army of bacteriophages and releasing it into the environment. In the lysogenic phase, it's a little bit different. It doesn't break down the bacterial chromosome. Instead, it incorporates itself. And then it starts replicating and replicating and further replicating, and eventually it will release related to some type of environmental stressor. There's two different types of transduction, so not only are there two phases, which is lytic and lysogenic, but there are also two types. So to make this simple, I'm just going to give you a little textbook definition, and I believe the name defines the function. So with transduction, again, you're taking DNA that's transferred from one cell to another via a replicating virus. Another name for a replicating virus is a bacteriophage. So when we have generalized transduction, this is some random piece of genetic material or DNA that's going from the donor to the recipient. The vehicle that does this is the phage, or the bacterial phage. So that is generalized transduction. It is a random piece of DNA from the donor being taken in by the recipient. The other one, or the other type of transduction, is known as specialized. This is when very specific host DNA sequences are transferred via the phage. Generalized is random, and specialized is a specific sequence of the host DNA is transferred via phage, and phage is a shortened name for a bacteriophage, which, please remember, these are actually viruses. In the picture, it's this guy right here. So with horizontal gene transfer, we've been talking about genetic materials passing back and forth between bacteria, causing all types of things such as antibiotic resistance. I mentioned the only type of plasmids that we're going to discuss in our class are F for fertility and R for resistance, specifically antibiotic resistance. There are many different kinds of plasmids, and if you think back to Unit 1, when we first started discussing it, actually it might have been the beginning of Unit 2, we had the bacterial chromosome, and then we had the plasmid, and together these two are known as a bacterial genome. So if you take a look down here, bacteriocins are also carried on plasmids. So when you say, what is a bacteriocin? It is a very, very specific antibacterial protein. Some strains of bacteria have it, and it's active against some strains of the same species and not others. So when you have things like antibiotic resistance, you have certain bacteriocins as well as other R plasmids that are passing back and forth between the bacteria. I would like for you to look at this statement that I'm putting the star next to, because this is going to come up on full blast in Unit 4, I believe. And this is, antibiotics do not or don't induce plasmids. So taking antibiotics don't induce them, but using antibiotics selects for strains that carry these plasmids. Somewhere write this down for yourself, because we're going to bring this up when we talk about antibacterial and antibiotic resistance. One of your case studies also deals with this topic. So this sentence will make perfect sense to you once we get a little further along. So outside of plasmids, we also have to add something known as transposons to the mix. So these are found in all types of cells, prokaryotic, eukaryotic, as well as viruses. The nickname for transposons are jumping genes. So these unique jumping genes, it's pretty much a DNA sequence that has its ability to change where it's positioned in a genome, and it has the ability to reverse mutations and alter a cell's genetic identity. So literally, a chunk of genes jumps back and forth. So you have a chromosome, you have a plasmid, and then there are transposons, which are jumping genes. If you take a look at the picture up here on the right, you have a gene, and a transposon inserts itself, which then causes a disruption in that particular gene. It's important to note that 40 to 50% of human DNA is not only a transposon, but a retrotransposon. Retro means going back. So it jumps in, and then just as quickly it jumps out. This is important to understand because if it doesn't have the ability to jump out, we can be sitting in a chair listening to a lecture and grow an ear on top of our head, essentially. So when you see that pretty multicolored corn around the fall season and the flowers with those odd colors, this is sometimes a transposon that wasn't a retrotransposon. So it caused a mutation because a transposon jumped itself into a particular gene. In Unit 1, you knew or saw on your exam that you had many, many pictures of the gram wall or the cell wall. Was it gram positive? Was it gram negative? And various questions about it. This is going to be that picture that appears a multitude of times for your Unit 2 exam. Why? Because everything we've just discussed in this PowerPoint is on this one single picture. This is a bacteria that is the recipient. Notice that there is an A, a B, and a C. A, you should be able to look at that after reviewing the PowerPoint we just went through and realize that this one is related to conjugation. And how do you know that? Because the two are touching, so there is a pili here. Conjugation is the only one where we talked about a connection between the two bacteria in that manner. Then if you take a look at B, you should be able to realize that this is transduction. And the hint is that it says transfer by viral delivery, and here is your bacteriophage. So this is your donor cell, and again, giving genetic material via a bacteriophage to the recipient. Here you have C, and the key word that I asked everyone to remember for transformation is dead. Based on the picture, you can see that this bacteria is disintegrating. This is all of the genetic material that has been released. So letter C is transformation. Expect to see this picture. What's happening at A and B and C? Where is the donor? Where is the recipient? Understanding generalized versus specialized transduction, where generalized is a random piece of DNA transferring via bacteriophage, where specialized it is a very specific segment of DNA being transferred from the donor to the recipient. By looking at this picture, I can also ask you what type of plasmid this is. So if you look all around this picture carefully, it says resistance gene right here. It says receiving resistance genes right here. It's written here. It's also written here. So this is an R plasmid, and the R is for resistance. Make sure that this picture is something you understand. You can label A, B, and C. You know what type of plasmid is being discussed, which is the donor, which is the recipient. Remember, in conjugation, there are two different kinds. This is regular conjugation. A second kind is HFR, where you have an incorporation and everything is occurring at 1,000 times the rate or speed of traditional typical conjugation. So please review this picture multiple times. If you are printing out your lecture outlines, there is a picture of this at the end of the outline as well.